First of all, my name is Haley Chiswick. I'm going to be the moderator for today's webinar. And I would ask that for those of you who are participating, if you have any questions about the talk as it occurs, please direct your questions to the chat. So at the end of the talk, we can go through and discuss them. And today I have the pleasure of introducing Dr. Steven Hicks. He is an associate professor at the University of Pennsylvania. He has done a lot of research in microRNA, including work in breastfeeding, in autism spectrum disorder in children, as well as, of course, concussion. And I truly believe the talk you're about to hear is coming from an absolute expert in this field. So I'm honored to introduce Dr. Hicks today. And Dr. Hicks, you can take it away.
Great, thanks Haley, and thanks everyone for joining today to hear a little bit about our work using microRNAs as biomarkers in concussion. So I do have a couple disclosures. I'm named as a co-inventor on microRNA related intellectual property that my institution has patented and licensed to a company called Quadrant Biosciences. And I do serve as a scientific advisory board member for that company, helping to kind of guide the use of our technology. So for today's talk, I'm going to discuss some of the potential utility of saliva microRNAs as biomarkers of traumatic brain injury, focusing specifically on mild traumatic brain injury or concussion. But I'm also going to identify some of the limitations in the scientific literature currently involving microRNAs that really need to be addressed before this can move on to clinical implementation. I'm a general pediatrician and a physician scientist, so whenever I think about a problem that I want to address through research, I think about it in terms of clinical gaps. And for me in concussion, there's really three large clinical gaps when I'm seeing patients in my clinic who have a concussion. The first is with diagnosis. So it's pretty well established that concussion is underreported. A study in 2015 showed that nearly a quarter of athletes report some pressure not to disclose their concussion symptoms. And as a result of that, concussion is generally underdiagnosed. So nearly a third of athletes have sustained a previously undiagnosed concussion. This stems in part because we are reliant on subjective symptom reports. A study from 2013 showed that at that time, the majority of athletic trainers were still not using neurocognitive or balance testing, although I suspect those numbers have improved in the last 10 years or so. A second gap is in our ability to provide prognoses. And for me, this is the biggest one in concussion. Here in general pediatrics, oftentimes patients come to me already with a diagnosis. And my job is to counsel families about what's going to happen over the next 2 to 4 weeks. And really, I feel that there's insufficient tools to do that. A study in 2012, which surveyed pediatricians, showed that most of them agree there's insufficient tools for management and family education. And I think this really lies at the heart of parental anxiety about what they can expect after their child's concussion. Even the CDC in its most recent guidelines published in 2018 admitted that although some factors predict risk for prolonged concussion symptoms, every child is different and it's really hard to tell who's going to have those prolonged symptoms. And then finally, a major gap for me is in decisions about returning to activity. So part of this stems from a problem called sandbagging, where nearly a third of athletes will tell you that they've purposely given submaximal effort on their baseline neurocognitive tests so that they can return to sports earlier. Not so much in an athlete population, but often to delay return to academics. There's some evidence that students will exaggerate their concussion symptoms to try and avoid or get out of return to school. So how do microRNAs fit into all of this, and what are microRNAs? So they are short non-coding nucleic acids that regulate protein expression. You can think of them as on-off switches that basically turn off the step that goes from a coding messenger RNA to a functional protein in your body. There's about 2,500 different microRNAs, and they each turn off a different set of proteins. So your body can upregulate or downregulate microRNAs in real time to change the physiology within a cell and how that cell is functioning. But also you can capture microRNAs outside of the cell, because many cells in our body package them into exosomes and then release them into extracellular fluid as a signaling tool to kind of coordinate protein expression within a cell network. One of the cell types that does this a lot is neurons. It turns out that microRNAs are really abundant in biofluids, particularly in saliva. The first study that I'm aware of that looked at microRNA levels in traumatic brain injury was a study by Riddell and colleagues in 2010. That study was a pilot study of adults who had suffered severe traumatic brain injury, and they identified a handful of microRNAs in serum that showed differences between the TBI group and healthy controls. But for me as a pediatrician, I was interested back in 2015 in how I could take that information and apply it to children or pediatric populations, and also do it maybe in a non-invasive way, because a huge hurdle to getting participants in our studies and then also getting families to agree to do tests is how invasive or how painful that test is. So knowing that microRNA is abundant in saliva, and knowing that most of the microRNA in saliva is contained within an exosome, we postulated that perhaps much of the microRNA in saliva is actually coming from neurons. And the reason for that hypothesis is that your mouth contains 5 different cranial nerves, and they're constantly signaling to one another by releasing microRNAs into your spit. So to look more closely at this hypothesis, we collected saliva from a healthy individual. We isolated exosomes, which are shown here in this electron micrograph. And then we measured the proteins in those exosomes. So the first 10 or so proteins shown here by densitometry are proteins that you would probably guess to be important in the mouth in terms of immunologic function. So most of those first 10 proteins there are involved in fighting viruses and bacteria that your mouth encounters on a daily basis. But the proteins that are labeled with the red star or the green star are specific to neurons and glia. And you'll notice that those proteins in saliva exosomes are actually as abundant, if not more abundant, than immunologic related proteins, which tells us that many of the exosomes in the mouth are actually coming from neurons or glia. If you're still having kind of trouble wrapping your head around how this is possible to collect brain information from saliva, I'd ask you to think a little bit about rabies virus physiology. So essentially, when a person gets a bite from an animal infected with the rabies virus, that virus, which is a single-stranded RNA virus similar to microRNA, travels up the dorsal root ganglion, up the spinal cord into the brain. And then the way it makes its way onto the next host is via retrograde axonal transport through the cranial nerves down into the saliva glands. And so that's similar to the mechanism that we propose for how brain-related microRNA makes its way into saliva after a traumatic brain injury. Really important for considering using this as a tool in your research toolbox is that saliva is really easy to collect. So it is collected in less than 30 seconds. It can be collected by participants at home themselves without any training. We usually provide them with a short 30-second video to show them how to do it. And then once they collect it, they can ship it to us via regular mail. And it's stable in the collection devices we use from DNA Genotech for 2 months at room temperature. And then once you get it into the lab, you can pretty easily quantify the microRNA in that sample using either high throughput RNA sequencing or PCR, which has immediate applications in clinical medicine. So our hypotheses, which we've tested in a number of studies over the last 5 to 7 years, are first that saliva microRNA levels are altered in a mild traumatic brain injury. And second, that those saliva microRNA profiles may be used to identify or diagnose children who have MTBI. We hypothesized that saliva microRNA levels may be able to predict the duration of concussion symptoms. And then measuring those microRNA levels over time may allow us to identify individuals who have recovered from their concussion. So one of the first studies that we did to test these hypotheses involved a sample of 50 mixed martial arts fighters. These are young adults in their 20s, amateur fighters. And we like to say that this is one of the few times you can actually schedule a concussion. Two fighters walk into a ring and typically one of them is walking out and the other one is being helped out after a brain injury. So in these 50 fighters, we assessed them longitudinally, first pre-fight, where we collected biofluids and did functional assessments. And then during the fight, recorded the number of hits to the head that each fighter received. And then again, 1 hour after the fight, 2 to 3 days after the fight, and 1 week after the fight, repeated biofluid collection and functional assessments. So the biofluids that we obtained from each fighter were saliva and blood. And then the functional assessments that we performed involved balance testing, several different tests with eyes open or closed and legs tandem or apart, and then neurocognitive testing, which involved spontaneous reaction time, go, no go, and procedural reaction time. So this figure shows how cognitive and balance measures changed in the fighters over time. So all the way to the left, you see baseline Z-score zeros for all these different fighters pre-fight. And then in fighters who had no hits to the head, these are either a fighter who landed a knockout blow before they even were struck, or in some cases, a fighter whose fight was canceled. You can see there's pretty minimal change in balancing cognitive measures. But 1 hour after the fight, the balance measures for the most part are going up. And that is actually a reflection of balance errors. So there's an increase in balance errors. And the cognitive measures are mostly going down. And that is a reflection of poor or worsening cognition function. 2 to 3 days after the fight, both of those measures start to recover. And then 1 week after the fight, you actually start to see some learning effects in the cognitive measures where they're performing significantly better than baseline. So if we look at how saliva microRNA changes over that time span, we can see that there's about 14 or 15 microRNAs that actually show a very acute response to the fight. So pre-fight, they're all at their baseline level. Fighters who had no hits to the head or had their fight canceled see minimal differences in these 12 microRNAs. And then 1 hour after the fight, there's a giant spike. So this is important information. It tells us that microRNAs are changing pretty rapidly after you've been hit in the head. It doesn't take days to weeks to see a change. There certainly are microRNAs that don't change for 3 days, 7 days, but there are also a handful of microRNAs that we call acute responders that are changed within an hour after being hit in the head. And most of those acute responders, you can see, go back to a baseline level 2 to 3 days after the fight and stay that way 1 week after the fight. In comparison, in serum, many of the changes we see are a decreased expression rather than an increased expression. And those changes tend not to occur until 2 to 3 days or maybe even 7 days after a fight. So this is one of the reasons that moving forward, we decided to focus specifically on saliva. We think that it provides a more acute response to traumatic brain injury, and that it allows us to maybe identify diagnostic markers that would not be possible in serum where things aren't changing for several days. So the next study that we performed was focused on children. And the purpose of this study was to show or identify particular microRNAs that had similar changes in the cerebrospinal fluid, or CSF, after a severe traumatic brain injury as they showed in saliva after a mild traumatic brain injury. So for this study, we enrolled 8 children who had a severe TBI and we're in a pediatric ICU. We collected 23 samples from those participants over 3 different periods. Day 1 after the injury, day 4 to 7, and day 8 to 17. We also had 3 controls who we got CSF from that did not have a traumatic brain injury. They were undergoing rule-out sepsis workups. And then for saliva, we collected 60 saliva samples from 60 different children with a mild TBI, as well as 18 saliva samples from 18 healthy matched controls. And then by looking at the microRNAs that changed in CSF after a severe TBI and microRNAs that changed in saliva after mild TBI, we could identify those microRNAs that were changing in the same direction in both biofluids after TBI. So in total, we identified, I believe it was 10 microRNAs in that study, and I'm showing you 4 of them here. So the control group is the dark box plot, and the TBI group is the gray box plot. The top row is showing you the change in CSF, the bottom row, the change in saliva. And so you'll notice each column is a different microRNA, and the change in CSF parallels the change in saliva. So here for miR29C, it increases in CSF after a severe TBI. It increases in saliva after a mild TBI. Whereas for miR26B, it's decreasing in CSF after a severe TBI and decreasing in saliva after a mild TBI. I'm just going to highlight these 2 microRNAs, miR30 and miR320C, because you're going to see those numbers kind of pop up again and again in our studies going forward. And so I just want to highlight the fact that these seem to be kind of reliable markers with utility for a lot of different diagnostic, prognostic, return to play kind of decision making. All right, so in this pilot study, right, which admittedly is small sample size, we've only got 60 children with mild TBI here. We wanted to know, could we take a handful of these microRNAs and accurately identify who had a traumatic brain injury and who was a healthy control? Not a particularly useful clinical decision-making tool. I can pretty much eyeball a kid in clinic and know if they're perfectly healthy or have been hit in the head, but it's proof of principle. So this is an ROC plot. For those of you who aren't familiar with it, the AUC stands for area under the curve. And a test that is perfectly sensitive and perfectly specific, the blue line would go straight up and then straight across, and the AUC would be 1.0 or 100% accuracy, so to speak. Spoiler alert, there is no test in clinical medicine with 100% accuracy. Typically tests with an accuracy above 80% are pretty darn good. So in this small pilot study, 5 microRNAs allow us to differentiate children who've got a mild TBI from children who are healthy with about 85% accuracy. To kind of put that in clinical context for you, if you or your children have ever had a rapid flu test in clinic, the AUC of that tool set, which is used pretty widely, is only about 0.6. So the next step for us was to, in this kind of pilot cohort, determine whether we could use the saliva microRNA levels to figure out which children with mild TBI were going to go on to have prolonged symptoms. So here for this study, we defined prolonged concussion symptoms as presence of symptoms 4 weeks after the injury, where the threshold was a symptom score of 5 or greater on the SCAT-3. In our cohort, 30 children met this criteria and 22 had acute resolution of symptoms. This is a skewed ratio, and it's because we were recruiting from our concussion clinic, which kind of enriches for kids at risk for prolonged symptoms. So this is a partial least squares discriminant analysis, or PLSDA plot. And the way this works is it plots all of the participants with a mild traumatic brain injury in a 2-dimensional space based off of all of the 2,500 microRNA levels in their saliva. All right, so each dot is a different participant. The blue dots are kids who had acute resolution of their concussion symptoms, and the orange dots had prolonged symptoms. And what you would expect to see if their total microRNA profile predicted prolonged symptoms would be a complete separation of these 2 groups. We don't see that. We see a partial separation of the groups. And that is because really it's not every microRNA in your body that's determining whether you have prolonged symptoms. It's just a handful of microRNAs. And so for each microRNA, we can calculate what we call a variable importance and projection score, or VIP score. And that score is basically telling us how good is that microRNA at pushing those two groups apart in two-dimensional space. And so these are the top 15 microRNAs on this PLSDA. I've highlighted again miR30E and miR320C for you. miR320C, you'll notice, has low expression in kids who have acute resolution of their symptoms, whereas miR30E has high expression in kids who have acute symptom resolution. So again, we can take 5 microRNAs from that list and make a test that differentiates the kids with prolonged concussion symptoms from their peers with acute symptom resolution with an AUC of 0.85. One of the things that we did in this study was to start to look and see, is that any better than what we could do clinically? So we compared the predictive value of those 5 microRNAs against the child's initial SCAT3 score. And that symptom score, as reported by the child, was only about 65% accurate. When we used the symptom score provided by the parents, it got even less accurate, which I like to say is just another example of how parents have no idea what's going on with their adolescent child. And then finally, we compared the accuracy to the 5P tool developed by Zemek and colleagues. This was a modified version because we didn't have noise sensitivity or balance error scoring data for these participants. But the accuracy of that tool in our hands for this small cohort was about 62%. And that's similar to what was reported in their initial paper. So obviously, a lot of work, although these are promising results, a lot of work needed to be done to validate it in larger sample sizes, collect more clinically relevant tool sets like balance and neurocognitive measures, and then also enroll a wider variety of participants from multi-center sites. And that's essentially what we've done in the last 3 to 4 years. So the remainder of the data that I'm going to show you today arises from this follow-up study that included 11 sites, Penn State, where I work, as well as Colgate, Marist College, New York Institute of Technology, Syracuse, Buffalo, Temple, wide variety with geographic distribution. So this study includes 538 participants. Their average age is 18 years, and there's a slight preponderance of males in the data set. Before we touch the data, we split it up into a training set and a test set. So we use the training set to identify the best biomarkers, to create predictive algorithms, and then we don't touch the test set until we have our algorithm set, and then we externally apply it in this naive set that we haven't looked at. The training and test sets include equal ratio of mild TBI participants and control participants. And the controls are age and sex matched to the MTBI group. Importantly, we also use controls that have conditions that mimic mild traumatic brain injury. So our controls include children with orthopedic injury or anxiety and depression, children or adolescents who've just finished exercising, so they have post-exercise fatigue, chronic headache disorders, and problems with concentration as a result of underlying ADHD. So we've moved on from healthy controls. And in the majority of these participants, we collect 3 clinical assessment tools. We assess their balance with a computerized clear edge system. We use the DANA tool developed by the military to look at neurocognitive function. And then we use the SCAT5 for symptom assessment. Okay, so the first study that we did with this data set was to look at diagnostic utility of non-coding RNAs in saliva. And so essentially what you have here in this ROC plot are now 2 curves. You have a blue curve, which is from our training set, which is always going to be the best performing model. And then a pink curve, which is from that naive test set. where when we apply the algorithm in that set, we hope that the accuracy holds up. And so now in this much larger data set of over 500 individuals, we're able to create a non-coding RNA model with just 4 non-coding RNAs that can still predict who has a concussion and who doesn't with 85% accuracy, and that actually drops just slightly to 82% in that test set. In comparison, if you used their symptom scores to figure out who had a concussion and who didn't, your accuracy would be about 88%. This is not surprising. This has been published pretty well in the literature that that's how accurate symptoms are, or maybe even slightly more accurate, because that's what we use clinically to decide who has a concussion and who doesn't. A better comparison may be to look at the non-coding RNA model versus neurocognitive performance, where they are essentially similar accuracy. And then again, for a balanced model, which also is in the mid 80s. Clinically though, right, we're not like predicting or diagnosing concussion in a vacuum. And if we're following the CDC guidelines, we're actually using symptoms plus some sort of objective test, like a neurocognitive assessment. And really in this data set, adding neurocognitive assessment to symptom scores did not really improve our ability to tell apart concussed and non-concussed individuals at all. But when you use non-coding RNA with symptom scores, you do get a bump in accuracy all the way up to about 93% for the AUC. That's slightly higher than symptoms balance. And it's even higher than kind of like a kitchen sink model where you throw together neurocognition scores, balance scores, non-coding RNA, and symptoms. So really best performance can be just with symptom assessment and saliva collection in this cohort. All right, the next thing that we wanted to look at in this giant cohort was how are microRNAs changing over time? What are their dynamics? And can we use those dynamics to predict symptom trajectory and then also identify concussion recovery? So what I'm going to show you now are the levels of 15 microRNAs listed here in a control group, and then also in our concussion group at several different time points. So 0 to 2 days after the concussion, 3 to 6 days, 7 to 14 days, 15 to 29 days, and then 30 to 60 days later. So here blue indicates a low level of expression at baseline in most healthy individuals. And then red is showing you that it has a high level of expression at baseline in most people. And you can see pretty much immediately after your concussion, you get a complete flip in these 15 microRNAs. So the ones that have low level of expression in non-concussed people go way up after a concussion, and vice versa for the other half. That same pattern persists about 3 to 6 days after the injury. But then right around day 7, when most people are starting to experience an improvement in their symptoms, we see this change in the pattern of these 15 microRNAs. And by 2 to 4 weeks after their injury, they're at least trending back towards what looks like a healthy control level of expression. And if you look down here at the bottom of the graph, you see the symptom scores. So average symptom score in our control group is 3. And in the 0 to 2 day group, it's 33. By this 2 to 4 week time point, their symptom scores have kind of returned towards a baseline score, and so have the microRNAs. But what about in the kids who are still telling us they have symptoms 2 to 4 weeks out? Do they have a different pattern, and can use that pattern to tell who's recovered and who's not recovered? And there's MIR30 again. So to address this question, we basically took 112 participants in our study for whom we had data at least 3 weeks or more after their injury. We used their SCAT5 symptom report to determine if they had recovered or not. And then we compared the ability of saliva microRNAs at that 3 week to 4 week time point to differentiate recovered versus non-recovered. The accuracy of the microRNAs shown here in green was about 83%. And that was significantly better than the accuracy of a model using both balance and cognitive measures, which was only about 73% accurate. If you combine RNA and balance and cognitive measures together, you get the highest accuracy. That's just a slight bump from 83 to 86%. So here in this cohort of 112 individuals using 11 RNAs, we can start to get a sense of maybe who has recovered from their concussion and is okay to return to play and who's not. So the most recent study, we actually just had this accepted in Journal of Neurotrauma this month, was to start to look at whether we could use saliva microRNA profiles to maybe define some biologic phenotypes of concussion. Why do this? Well, for starters, right, there are a lot of different medicines that have been tried to help individuals with concussion, and they don't universally work. And perhaps that's because there is a lot of different biology going on in individuals with concussion. No two people maybe experience their concussion in the same way, although there are patterns that different people experience. Right, concussion is a very diverse kind of constellation of symptoms. So rather than relying on symptom reports to kind of divide up our patients into phenotypes, maybe we can actually use microRNAs to divide them up by biologic response. So we took the 251 concussed participants in our study, and we used a k-means clustering approach and used the elbow method to determine what the optimal number of clusters was within our cohort. Turned out it was 10 clusters, and you can see those clusters here on this PCA model. If you take those 10 clusters and then you start to compare them in terms of their symptom reports, we do find that the 10 different clusters have significant differences in reports of fatigue, memory, confusion, and sleep difficulty. So this heat map here, each column represents a different biologic cluster. And then each row represents a different symptom on the STAT5 symptom report. And so what you can kind of notice right away is there are two clusters, cluster 4 and 7, that have really high levels of symptoms, and then two clusters 3 and 5 that have really low levels of symptoms. There's also this really interesting cluster 10, where the majority of their symptoms right here in the middle are coming from what I would consider to be like emotional symptoms. So they endorse feeling more emotional, feeling sad, feeling nervous. And so actually their microRNA profiles are helping to differentiate all these different groups. If we look at the pathways that the microRNAs that define each group are targeting, there are actually 16 different pathways being targeted. And not surprisingly, they include pathways that have been reported as being important in brain injury and brain recovery in animal studies and cell studies, and even things we think about pharmacologically when we're treating our patients. So the microRNAs that we used here target adrenergic signaling, GABA signaling, estrogen signaling, fatty acid metabolism, and TGF-beta. Oh, and then these are just the 4 symptoms that differed between the phenotypes. All right, so far I've painted a pretty rosy picture about how great saliva microRNAs are and how easy they are to use and all this great promise that they hold. But I would be remiss if I didn't also talk about many of the things that can confound saliva microRNA levels that are really important to consider if you're going to use them in your study or tack them on to maybe other functional measures that you're doing. So the first is that a large number of saliva microRNAs are affected by collection time. That is, they have diurnal oscillation. So actually, of the 2,500 microRNAs that your body expresses, almost a quarter of them change with the time of day. And we know this from a study that we did about 4 years ago, where we collected 120 saliva samples over the course of several days from 9 healthy individuals. Here's an example of one of those microRNAs and how it has really low collection times first thing in the morning. Those levels rise throughout the day. And then at night when the person's sleeping, start to dip again. So how do you handle this knowledge, and how can you use it to kind of improve the accuracy of your research? Well, the first is that you want to make sure you're matching collection time for your TBI and control groups. Because if you're getting all your controls from a well child clinic where they come for visits between 8 and noon, and then you're getting all your TBI participants from an emergency department where they come in between 6 at night and midnight, you're obviously going to have 25% of your saliva microRNAs that look different between the two groups. And it has nothing to do with a concussion. It's just because you collected the samples at different times a day. So you really need to match your two groups in terms of collection time. The other approach would be just to exclude the 1/4 of microRNAs that have effects of time as potential biomarkers, although you're giving up a large potential swath of maybe concussion responsive biomarkers when you do that. A third approach is to include collection time as a covariate and help control for that when you're building predictive models. Another factor to consider is food intake. So to look at this, we collected 116 samples from 58 healthy participants when they were fasting, when they woke up first thing in the morning, and then 15 minutes after they ate breakfast. So what you can see here on this PLSDA is that we actually do get complete separation of fasting samples in red from postprandial samples in green. And at first glance, that's perhaps concerning, but actually it turns out if you look at the percent variance contributed by each component here on the PLSDA figure, you'll notice it's only 2% and 2%. And what that means essentially is that really, it turns out only about 4% of all microRNAs are affected by food intake. So here it's much easier just to take those 4% of microRNAs and exclude them and say, we're not even going to consider those as biomarkers. You're not giving up a large percentage of microRNAs by doing that. In concussion studies, you can't really ask your participants to be fasting because you want to get the sample as soon as they come in after their head injury. So you don't want to make them then wait 3 to 4 hours to give you a sample. Another approach is just as with collection time, you can ask them, when did you last eat? And then use that as a covariate in your models that you build. And then the last, maybe most salient confounder that we need to consider in concussion is exercise, right? Because at least in our cohort, over half of our concussed teenagers are coming to us after a sports-related concussion. Many of them have just finished exercising. So to address this issue, we collected saliva microRNAs from 83 athletes. These are athletes involved in both contact and non-contact sports. Right before and then right after they had exercised in practice. And then we looked at microRNAs that changed with exercise. We focused at this point specifically on microRNAs that we had published previously in concussion literature. And we identified 2 that we really needed to exclude going forward because they do show a fairly large impact of exercise. That's MIR 532 shown here and MIR 182. We also looked at microRNAs in 106 athletes where we collected a sample at the beginning of their season and then again at the end of their season. So why would we do that? Because if you're going to start to use this technology to collect preseason baseline and then repeat a sample after an athlete has a suspected concussion, you want to know that the microRNAs you see changing are because they just had a concussion and not because maybe they now have enhanced exercise capacity or muscle-related changes that occur with 3 months of conditioning. So with that, we excluded another set of microRNAs. We were actually able to whittle down our predictive model to just 2 microRNAs, MIR 27A 5P and miR30A3P, which you see here on the bottom right corner. And that 2 microRNA predictive algorithm had about 81% accuracy in figuring out who was concussed and who was not concussed. And most importantly, there was no increased rate of inaccurate concussion calls among our exercise controls. And there is no increased rate of inaccuracy among sports-related concussion versus non-sport concussions. The last barrier that maybe doesn't need to be addressed for prognostic testing or return to play testing, but I think is important for diagnosis is the sample processing time. So I mentioned earlier that you can use PCR to measure microRNAs. It's the same technology we use for COVID testing. So many of you are probably familiar with the 4-hour turnaround time that is typically seen with a PCR COVID test. You can expect maybe something a little faster if you had a portable PCR machine and ran a sample right at the bedside, you could get a 2-hour turnaround. But I think most emergency departments are going to want to see something on the order of like 20 to 30 minutes instead. So here at Penn State, we've been working with molecular engineers in our Department of Engineering to kind of advance the way that we can measure microRNAs at the bedside. And we're doing this with something called rolling circle amplification. So we published this paper in Analytical Chemistry just a couple months ago, showing that with this rolling circle technique, we can accurately measure levels of 3 concussion-related microRNAs directly from raw saliva. And then also that we can do that in a pretty time-sensitive manner, less than an hour. All right, so what is the future of microRNA research and application of this technology maybe in concussion patients? Well, I think first of all, we need to continue to validate these findings in larger and larger sample size. And those large sample size really need to include multi-center collaborations. It's going to be important that studies use appropriate control participants. I think we're beyond the point of using healthy controls. We really need to be using naive test sets to validate any markers that we put forward. And if possible, validate those markers using the most rapid possible measures. At the end of the day, anything that we do with microRNAs needs to be an improvement on what's available clinically. So having clinical tools in your study to compare against microRNAs like balance testing or neurocognitive testing is, I think, really critical. And then, as I just showed, there are a number of potential confounding factors that you really should be collecting if you're going to collect saliva and measure microRNA in your studies. So in conclusion, I think that we've been able to show pretty convincingly that saliva microRNA levels do change in individuals who have had head trauma, and that the microRNA profiles in mild TBI do to some extent mirror the CSF microRNA profiles you see in severe TBI. You can use those microRNA levels to aid identification of mild TBI. I would never presume that microRNA is going to be a tool that's used in isolation. I see it as one more tool in a toolbox, hopefully in the next 5 to 10 years. And then maybe not just a diagnostic tool, but also a tool that we use to provide prognoses for our patients, and then also support our decision-making regarding return to activity decisions. So with that, I will wrap up the talk. I would be remiss if I did not acknowledge the many individuals who have contributed to this work and our numerous funding sources, including Children's Miracle Network and the NIH. So thanks for your attention and happy to take questions.
Yeah, thank you so much, Dr. Hicks, for that extremely informative talk. I do see a question in the chat, and if there are any other questions, I direct everyone to put them in the chat, and we'll go through a couple as time permits. I also wanted to let everybody know that there is going to be a recording of this available to you in a couple days after the talk has been completed. So just to start off, I'm going to go ahead and ask a question here, as some people are utilizing the chat function. But you mentioned in the beginning of your talk that this might be a potential useful adjunct for clinicians. And you also mentioned that there are changes diurnally and changes, from time to injury. So for a clinician who might be naive to the use of microRNA, what are some of the key points and key factors that they should know about this collection to add it into their clinical toolbox?
Yeah, it's a great question. So first of all, I think that one of the great things they should know about it as a plus is that you can use it to do longitudinal collections, and you should do longitudinal collections, because as we showed, the things that change an hour after a brain injury are not the same things that are changed 3 days, 7 days, 14 days after the injury. And it's important to have a good sense of kind of the longitudinal patterns that you see in your cohort or in your patients. So I would encourage people who are thinking about using the technology to try and use it longitudinally. And that can easily be done just by swabbing the person when you first enroll them and then sending them home with swabs to collect at home. But we always collect 3 important pieces of information alongside every swab we collect. The first is what time of day it was collected. The second is when the person last ate. The third is when they last slept. And the fourth is when they last exercised. And what we feel is that knowing what we know about microRNAs that are really impacted by those activities, we throw those out right off the bat. And then anything else that has just really mild impacts of those activities, we can control for it by potentially including those times in the models that we build, the predictive models. So I would just say, use this longitudinally, and then also collect those 4 pieces of information, and you should be all set. Also, use an RNA stabilizing kit, because there are major impacts of just collecting raw saliva and trying to get it in a freezer versus like using RNA stabilizer, where you don't have to worry about time from collection to time that it hits a -20 or a -80 freezer.
Yeah, absolutely. That's phenomenal advice. There is one question in the chat that is a bit lengthy, so I'm going to try to just shorten it up here. But there is a researcher who is studying animal-assisted therapeutic interventions, specifically looking at pathways, including cortisol and cytokines, and is interested of the utility of including microRNA in their collection, as well as some other confounders that they might want to be concerned with in this animal model.
Yeah, I would say that there definitely are studies looking at microRNA levels in animal models of TBI. We have not done any of that animal work, but there is a lot of between species conservation in microRNA sequences. So microRNAs that change with TBI in a human, I would expect to have large amounts of overlap of what you see changing in an animal model. If you're already collecting the saliva for other purposes, it should be pretty easy to measure microRNA out of it. Saliva, as I mentioned early in the talk, is pretty concentrated source of microRNA. So you don't need large volumes to get out the amount of RNA necessary for PCR. So I would encourage that as a adjunctive measure in any study. And for anybody on the call who is interested in using it, happy to correspond via email and provide you with some guidance or answer questions you might have on how to do that.
All right, great. And moving along to the next question from Brennan Jenkins. They are curious about evidence to suggest saliva microRNA testing on patients who have previously sustained a TBI, maybe a couple months or a year ago. And I'd like to add on to that, not just a severe TBI, but what is the effect of concussion history and mild traumatic brain injury on these microRNA panels?
Yeah. So we actually published a study that I did not show the results of today, where we had a cohort of retired Hall of Fame NFL players and an age-matched cohort who did not have a career playing football. And we used those 2 cohorts to identify microRNAs that differed, and then looked to see if those microRNAs were correlated with the number of concussions or concussion-like episodes that the Hall of Fame football players told us they had had. So we were able to identify there a list of candidates. And then as a way to sort of externally validate those markers, we took a large group of, I think it was over 200 college athletes who we had saliva samples and concussion history from. These athletes had all sustained their concussions at least 3 months earlier and had fully resolved symptoms. But we looked to see if there were correlations between the number of concussions they reported and these same microRNA candidates that we had identified in the Hall of Fame football players. And I think we landed on two that were the same, showed the same patterns across both age groups, and think that perhaps those two microRNAs could be markers of past concussion, or might have be impacted after a concussion and then have a sustained kind of dysregulation that goes on for years after the concussion. I cannot recall what numbers they were off the top of my head, but that paper is out there. It's open access, and it's yeah. So check that one out. I'm happy to, if I can send it to you, Haley, and you can share it with whoever in the group wants to see it.
Yeah, sure. I will make a note of that. Moving on to the next question we have here, Roger Zemek is interested if your studies of MTBI exclude participants who had any structural neuroimaging changes to differentiate concussions from other subsets of mild traumatic brain injury.
Yeah, so we do have that as an exclusion criteria in our studies. We don't image every participant, but if a participant comes in and gets imaging as part of the clinical workflow, and then they are found to have some sort of structural change or brain bleed, they're excluded. The exception would be in that first study I showed where we had a severe TBI cohort. There are some obvious structural changes in that severe group, but all of the validation work I showed in the large cohort of over 500 participants, anybody with a structural change is excluded.
Yeah, absolutely. So a follow-up question as well. The majority of your talk today kind of looked at microRNA, but there are other small non-coding RNAs, and you touched on that a little bit. But the question is, when is microRNA potentially a more applicable biomarker than other small non-coding RNAs? And how do you kind of parse between the difference between small non-coding and microRNAs, which might be more useful?
Yeah. So there's one other group, to my knowledge, that's doing this similar kind of work on a large scale. There's plenty of other groups doing this in pilot studies, but that other group doing it on a large scale is out of the UK, and they're doing this in their professional. They have access to all the professional rugby players over there, and their paper in the British Journal of Sports Medicine, which was published I think about 1.5 years ago, used microRNAs along with some other types of non-coding RNAs. These would be things like Pee-wee interacting RNAs, small nucleolar RNAs, and they achieved pretty high accuracy in doing that. We actually also have started to include some of these small non-coding RNAs that are not microRNAs in our own work. They do bump accuracy when you start to include other RNA types. The problem, or my reservation in using them, is that their functions are not well understood in the way that microRNAs are. So I can take a microRNA, look at its sequence, and then basically tell you which messenger RNAs or proteins it's probably going to turn on and off. But for many small nucleolar RNAs or Pee-wee interacting RNAs, I have kind of no idea what they're doing. To be honest, I know what they do as a class of molecules, but we don't understand on an individual molecule basis what they each do. And so it's hard to weed out things that may be statistical noise from actual biologic function. So I would just be careful about building predictive models using molecules that you don't know what they're doing on a biologic level. So for that reason, We'll include a few, but mostly we focus on microRNA.
All right, I'm going to ask one final question here. And if there are any other questions that maybe we can do quickly as we're kind of wrapping up here, the chat is still open for those of you who might have a last second question, but I'll go ahead. Actually, I see one other question that just popped in, so I'll ask that one instead. Another question from Roger Zemek. What do you envision as the timeline in obtaining FDA regulatory approval as a test? How many years away are we from being able to use this clinically?
I think best case scenario, probably 3 years. And the reason that I put that out as the timeline is, so right now, I've been working with a company I mentioned at the top of the talk called Quadrant Biosciences, who's licensed the technology from my institution, Penn State. And Quadrant Biosciences has a large NIH R42 grant to validate these markers. It's a 3-year grant, and now it's about almost a year in. And so I think they'll probably, if that study is successful, they would take the results, go to the FDA, and then have approval. So that's why I kind of throw out 3 years as a rough ballpark for how far away this might be.
Mm-hmm. All right. Well, thank you for answering those questions. I don't see any more questions in the chat. But yeah, I think we're good to wrap up a little 2 minutes early, but thank you all for coming to the talk today. And if there are any additional questions, I'll be around until 12 o'clock. It looks like Nick has a quick question as well.
More just big thanks, Haley, and to Dr. Hicks. So as our co-chair of our IBIA Concussion Special Interest Group, what a fantastic presentation today from Dr. Hicks. Huge thanks, and Haley, great job moderating, and big thanks to everyone for joining us today and asking such great questions. I'll just put a little plug to join us again at our next webinar session for our Special Interest Group, which will be on Wednesday, May 25th, 11 a.m. Eastern Standard Time, and we're going to hear from Dr. Mike Ellis from the University of Manitoba, who'll be sharing on his amazing work specific to telemedicine for concussion care in remote and rural communities. So please join us again. And Haley, over to you for closing words.
Yeah, absolutely. Big thank you to everyone who joined us today. We look forward to seeing you guys at the next talk. And thank you all so much. We'll see you all later.